Okay, so I'll spend my time until the presentation is open and say thank you for Mira for inviting me and for organizing this very stimulating event. I'm always the last, so it's good. I think it's a good sign. I want to talk about many things, but I'm going to be precise. So basically, you know, last November, I had this early morning with Ian Reid, the CEO of Pfizer, and we talked about the lack of innovation in pharmaceutical industry. So there is really lack of innovation in pharmaceutical industry, and I think that one of the things that we are experiencing is that people are, you know, they're stopped thinking about how to innovate things, and everyone says always, you know, let's go with the stream. Right? So let's do something that everyone agrees. It's very easy to say no, right? But you need a lot of gut to say yes for a new idea. So how do we define our challenges? I'm going to start with a very simple quiz, so bear with me. I'm sure some of you knows this. Let's assume a car has fallen into the river, and now your task is to take it out from the river. Everything I'm going to tell you are true stories. Okay? This is from Aarhus, from Denmark. So this is, and it's been around in the internet for quite a lot of time. So if you know that, don't destroy the others. A car is falling into the river, you need to take it out. What are you doing? That's one option. Another option? Drain the water. That's a very smart, but it's a big one. Okay. So 95% of the people will probably say, you know, bring a bigger car, right? This is the example, right? Very, very classical. This is a lake or a river, actually it's a sea, and this is the car, and they brought a bigger car, right, to take it out. But only very, very sharp people or physicists will now notice that this car, small one, is full of water, right? So the momentum that this guy calculated very efficiently is not the one that can handle this. And what will happen is this. Now we have two problems in the water, right? Instead of one. The solution is classical. We keep on thinking. It's the same thinking process. What are we going to do? A bigger car, right? And again, it's the same story. Okay. So now I want to show you a short film, a very short, and I need your help. I want to show a very quick minute what happens if you do have innovation, how the public actually take this into account. סרט, סרטון. כן, אני חושב שיש לך את זה פה בכרום. בלינק בכרום, כן. Okay, just open it in a wide stream. Very known, everyone knows this. 1994, the Today Show.
That little mark with the A and then the ring around it. At. See, that's what I said. Katie said she thought it was about.
Yeah. Oh.
But I've never heard it.
Around.
I've never heard it said. I've always seen the mark, but never heard it said. And then it sounded stupid when I said it. Violence at NBC.
אפשר להוריד את הווליום?
The lunchroom.
See, there it is. Violence at NBC, GE com. I mean,
What Allison should know.
what
do you
is
think about
internet
Allison?
anyway?
Internet is that massive computer network, the one that's becoming really big now.
What do you mean? How does one? What do you write to it like mail?
No, a lot of people use it and communicate with. I guess they can communicate with NBC writers and producers. Allison, can you explain what internet is?
No, she can't say anything in 10 seconds or less.
Oh, Allison will be in the studio shortly.
What is it?
it mean?
It's a giant computer network made up of started from.
Oh, I thought you were going to tell us what this was.
It's like
We're looking at the
computer
dictionary.
billboards.
It's not. It's a computer billboard, but it's
Right.
nationwide. It's several universities and everything all joined together.
Right,
And others can access it.
right.
And it's getting bigger and bigger.
It
Just
came in
right.
really handy during the quake. A lot of people, that's how they were communicating out to tell family and loved ones they were okay because all the phone lines were down.
I was telling Katie,
But you don't need that. You don't need a phone line to operate internet. No.
Okay, guys, so thank you. Basically, you know, this was a very interesting discussion. about what is the internet in 1994. So there was a nice innovation, but nobody understood it. Do we need it even? So I'm going to try to convince you now that we are on the verge of really going into bigger and bigger things, but the public need to try and understand this. So we need to communicate with the public much better than now. The end of the Today Show was that this guy in the middle said, I'm not going to put a dime on this because it's not important, right? So you can watch it. I didn't want to show you all of it. So we need to think out of the box, and these are all nice cliches, but at the end, we are really in a big revolution, and you know that each one reacts differently to drugs, and the classical example is that if you have a headache, one Tylenol is enough, the other one needs maybe 2 ibuprofen, and the third one will need a shot of whiskey, and everything will be fine. So we really interact differently to drug, and one notion is a genetic notion saying our profile is different, our genomic thing is different, we get lots of data, and I'm going to give you lots of numbers, you know, for sequencing. This was 1994, a dream, almost $1 billion per one human genome. In 2014, around 2,000 for one human genome. It takes about 48 hours then. Now we can do it with around 24 hours, right? Cheaper, around $1,000. The question is, what do we understand? I'm going to give you a personal example. When I started my career, I decided I'm going to do 2 things. I'm going to sequence myself, who knows what will happen, and then also I'm going to do a karyotype. In my karyotype, the first thing that was noticed that in chromosome 3, I have an inversion. What does it mean? Actually, it's not detailed in the literature. So back then, because it wasn't detailed in the literature, the first thing you do is to try to speculate. Is this the reason I was really bad at math? Or I was maternally imprinted to become a scientist? Or that I will have a disease that was not really still invented? All the answers are probably correct. I want to touch bases back with this because I want to mention that the first thing my oldest boy was born, I actually looked at him as well, and he was fine. Then the second one, and he was fine. The third one, he was fine. So this is a random issue which I'm still considering to explain how come, and it's correct, because I've done it a few times. So what does it mean? I don't know. We'll see later. Okay. So what can we do with all the information of the sequencing? Classically, we now know that in cancer we can sequence cancer patient in a particular disease, learn about the mutation and genes, and look at the expression, and then design potentially new drugs based on the sequence or in the RNA, and hopefully feed them back to the patient. There is one company, I'm not going to mention lots of names, one company is doing it already in Germany very nicely. Within about 3 weeks from sequencing, they're going back with neoantigens, kind of a vaccine approach, but it looks very promising. And then also you need the carriers, the delivery vehicles that Mauro talked about them, you know, and this is a very simple drawing, very simplicity on a cell. This is the messenger, a ribosome that will translate it into a protein that have a 3D structure, et cetera, and that in the end of the day, we can today silent gene, do it quite efficiently. We cannot really good yet upregulate proteins like with a modified messenger, but there are many people who are working on this, so it looks like another way. And of course, editing the genome, which is something very interesting, and another hype that will probably plateau, and at some point in a plateau, it will even create new drugs. So I'm a bit optimistic. Okay, so playing God with genes is great. And basically, the technology is here or will be here in the next, I would say, 5 to 10 years. So it's not big vision. But what we are still lacking is specificity. I think that all the vehicles that we are working, Mauro is working, and many other people in the world are working, are lacking a very, very precise address. And this address, this specificity, this GPS address that we can go on and hone into is missing because of lack of disease signatures. And there's so many types of disease signature, but we don't really understand. And I think here we need the computational people to come back to us and help us with this. So, okay, so this is something we are trying to have. You know, we have the drug. Let's assume it's an RNA drug. We have a vehicle. In this case, it's a liposome or a lipid-based nanoparticle. We have the targeting moiety, which is a GPS. And then, can we do it in a blockchain manner? What is blockchain? I tried to use all the buzzwords I could, actually. So blockchain is actually a strategy, okay, to secure money in transaction, international transaction. Not only money, also information. And actually, very important, genomic information and transcriptomic information. And it's been used now widely by several institutions around the world in order to do this in a secured manner and in different places around the world. So we are trying to analyze data, I'm going to go into this in a second, and basically transfer this through kind of a blockchain technology. So our specific lab is interested in leukocytes-implicated disease. We believe these are the best delivery vehicles because they can actually hone into different areas, etc. We are very much into inflammation, mostly IBD, bronchiolitis, and hematopoietic, mostly in cancer, mantle cell, etc., and some viral infection, and also in solid tumors that have immune activities. So, and this is the lab is divided into two major groups, people that are working in the drug delivery and generate monoclonal antibodies, also protein engineering, and people that are doing some exome and transcriptome analysis. And of course, the ideal situation is to combine those two into tailoring something very specific. Okay, and I'm going to talk a little bit about the RNA delivery carriers that have special requirement for leukocytes. Now, if you look at this, this is an illustration of a T cell. And you can see the majority is a nucleus. And if it's an activated one, so still the majority is a nucleus. So you don't have a lot of room to work with. So, of course, efficient encapsulation and specificity is true for any delivery system, and evading clearance mechanism, and avoiding toxicity and carrier internalization and endosomal escape. Everything is true. But these cells are more resistant to transfection. Actually, if you take subtypes of T cells, it's even more challenging. If you take B cells and memory B cells, even viruses cannot penetrate into those cells. And the question is why? So I'm going to start by giving you some examples. The first one is an inflammatory bowel disease. We have been working on this for many, many years. So cyclin D1 is aberrantly, it's a gene that's responsible for proliferation of both normal and malignant cells. And we ask a very specific question about why this gene is dramatically upregulated in leukocytes. So we set up a strategy to target systemically gut mononuclear leukocytes and selectively silence cyclin D1 in an experimental colitis model. And this, unfortunate, was published in this journal. Okay, so it's not bad. It's not bad. I have others, but this is not bad. Okay, so that's a bit old, 2008. But this was the first, it was a seminal paper, and something good came out of this paper. Actually, what became of this paper is a new drug. So we also generated 24 clones of anti-beta-7 integrin, which actually block leukocytes homing into the gut. And the eventual response was, I'm not going to show you the data, it was published, not important. What is important is that there is a new drug by this small company named Genentech that took this, and this is an anti-integrin antibody that it's basically a conformational specific antibody. It's one of the versions. Actually, the funniest thing, it was the worst version, the worst clone that was made is the best one in the humanization, but this has always happened, you never know. And this blocked the homing of lymphocytes into the gut. So something good came from a seminal paper. Oops, sorry. Okay, now staying with mantle cell lymphoma, stay with cyclin D1, okay? So I want to talk a little bit about mantle cell. Mantle cell is a very unique disease. It's quite rare, about 6% of all non-Hodgkin's. And it's a B-cell lymphoma, and it's considered to be incurable. although there are new pathway-dependent small molecules now that extend dramatically the survival to around 5 to 7 years. So it's still the poorest among the B-cell lymphomas, but it's still very promising. But it has a very unique genetic hallmark. About 85% of the patient experience a translocation of the gene that encodes for cyclin D1. So basically, we can design a specific approach to knock down this gene that is now under a very strong promoter and start to produce a lot of cyclin D1. And this is exactly what we have done. We first collaborate with the Broad Institute in order to sequence 80 patients that reveal 2 transcripts. One is a long messenger and one is a short one. And we design a kind of a personalized approach. It's not personal for the patient, it's personal for the mutation or for the translocation. And now we can knock down it dramatically. And this was done in collaboration with Alnylam Pharmaceutical. The work was done by Dr. Shiri Weinstein in my lab. And basically, she screened more than 500 sequences that was optimized after bioinformatics. And she revealed one of them. Just imagine how depressing it is, but one of them is really a potent siRNA. And it can knock down the gene dramatically in a picomolar concentration in the IC50 level. So it took a few years, and we have found a sequence. Oops, sorry. And then, oops, I ran a little bit too fast. And then we said, okay, let's now encapsulate this, and let's show that this can go into specific cells, in particular in these cells. And we have done this. We had to develop a model for mantle cell lymphoma. This was published last year in PNAS, 2 years ago actually now, if I think about this. And then we also wanted to choose a receptor target. It needs to be fast internalizing when you have those nucleic acid. And we chose CD38 as our target with an anti-CD38 antibody. And these are the results. Basically, I'm not going to show you all of this because, again, these are all published. But one of the things we have done, we have streamlined the process. So we can actually do now 1 liter in 1 hour. This is more than the industry can handle. 1 liter in 1 hour. Microfluidics, and then we have those antibodies on the surface, et cetera, et cetera. The only problem with this strategy is that you cannot control the orientation of the antibody. It's just chemical conjugation. It's very, very problematic because then you have different creatures, right? Some of them has more antibodies, some of them has less antibodies, some of them do not have antibody on the surface at all. How we can control this? So we are thinking about this in the past few years. Okay, I'm just going to show you quickly the result. This works very well, published, and at the end we have shown that, even therapeutically in a blinded experiment in Charles River, which was very nice because then you have another validation, you basically see that, it's not enough to block a single gene, but you can extend the survival, and also you have some biological activity from the antibody itself with a control group. Okay. So again, we can scale it up. We can go to your non-human primate. We can show that the liver enzyme looks fine. Everything is nice. So one of the things, the platform is flexible. You can do many other things. There's another science paper. Okay, so the motivation is that chemical conjugation is a problem. It's a scientifically and technical problem. How can we solve this technical problem? So, antibodies, as an example, are not all alike. Okay, they have different glycosylation, or they don't have. It depends how they were prepared. Actually, you can now have Fc exposure, so it's poor binding, and it can be uptake by many cells that are not the cells that you want them to be uptake. And, conjugation on globular surfaces, although chemistry is very nice, is non-controllable. So we decided to turn into biology, very simply. Biology has sometimes good solutions. One of them is just affinity, but you need to find the high affinity one. So we created the linker strategy, which is based on affinity. You're now creating the same linker that will be able to bind specifically to any antibody from the same class or isotype. okay? And then you can control the orientation. So in principle, you know, you need another component, which is when you inject something systemically, you need to avoid mononuclear phagocytes, macrophages, monocytes, DCs, etc., even neutrophils. So if this is a particle, you need to decorate it with some delaying agent. Mostly we use polyethylene glycol, okay, which is this guy. Okay, so this is an example of the brush like a polyethylene glycol. But also, you need to orient your targeting moiety. And this is, you know, when you have a blend, two blends like this, it's very hard to cut here the baloney. But if you have an oriented one, it's going to be perfect. So this is exactly what we designed, this linker strategy. And basically, we are using ionizable lipids. We're encapsulating nucleic acid. And we put on the surface this engineered antibody. And I'm going to show you how by a simple mixing with different antibody, you go to different targets. So we designed a mouse anti-rat IgG2A, I'm going to show you, and then a mouse anti-human and a human anti-human. We started with a complete antibody, turned it into a single chain, and then lipidated it in one transcript and purified this in a very special condition that there are minimal endotoxin on the way. I never say zero, because always there is some. And this is basically going to appear in Nature Nano very soon, in about a week or two, so you're the first to see this. And this is the design of the strategy, the plasmid. This is how we package this. Okay, we have particles, so we have nucleic acid on one side, our special lipids on the other side, microfluidics, we have these. We create this single chain lipidated and post-insertion strategy that goes into the membrane very, very nicely. Actually, we can do it also on the microfluidics. And then any antibody you will put will bind very nicely. And this is a very nice, you look at an SPR, a surface plasmon resonance. It has nice affinity, looks very, very nice. This is the schematic illustration. You can look at this or the linker here. This is the single chain that goes outside the particle. And now all I have to do is to convince you that this is a great strategy. So it's very versatile, okay? I'm going to show you. And it's protect the FC. So for this, I don't have time to show you, but basically, compared to chemical conjugation, you get much less FC portion. But this targeting versatility, I'm going to show you two examples. One of them is with lymphocytes. These are, let's say, all lymphocytes. So integrin beta 7 is expressed both on B cells and T cells. And then you have specific markers. CD3 is all T cells, CD8 is cytotoxic T lymphocytes, CD4 helper cells, CD25 is a subset of CD4. Some of them call them T regulatories, depends. And what we have done is to encapsulate, here in this case, siRNAs that are just labeled with Cy5, injected into mice. Hour later, take the spleen and gate them and see where they're going. And you can see here very nicely that we can go very, very specific into CD25. It's all in vivo compared to, let's say, monocytes or B cells or CTLs, et cetera, et cetera. So it's very specific. Can we see silencing with this? Yes. So we use the antibody entrolizumab, this is the Genentech antibody, an anti-beta 7 integrin that binds both CD8 and CD4. And also, in parallel, a very specific antibody against the CD4. And we just mix them, and this is what we expect. CD4 will enter only CD4, whereas where the beta 7 integrin will bind both of them. And we wanted to see the silencing, so we did the same experiment, injected into mice. Splenocytes were isolated 5 days later. And this is the silencing of CD45. So what you can see here is an anti-beta 7 integrin on the surface actually silence CD45 inside siRNA against CD45 in both CD4 cells and CD8 cells, while the CD4 only silence CD4 with nothing in the CD8. So it's very specific. Can this strategy become therapeutically relevant? We use the two models that you already know here, so the colitis model and the mantle cell lymphoma model. In this case, we use an anti-Ly6C, because in colitis, mononuclear phagocytes like macrophages There is a subset that are very, very important that express the slice 6C marker. And we have shown that by just 3 injections IV, we can get very nice scoring, and we silence very dramatically the expression of TNF when we entrap siRNA against TNF-alpha, and you get therapeutically relevant results. So it's less, you get less bleeding, less, the colon is better, the size, you look at the colonoscopy, it looks better, and also the other markers. Then we did the same thing with mantle cell lymphoma. Same experiment that you know before, but this time using siRNA not against cyclin D1, again another cell cycle regulator, and an anti-integrin beta-1, which is CD29. Here you can see basically the same trend. This is cell cycle arrest. But the interesting fact is that in order to perform this experiment, we need 250-fold less antibody. So this is a dramatic improvement in the potential to translate this to the next level. So what is the relevant to cancer, for an example, or for IBD? Basically, this linker may enable personalized medicine. Just imagine this diagram that you have different nanoparticles approved with different cargos, with the linker on them. We have 41 FDA-approved humanized antibody. You just need to mix them very quickly near the patient. The incorporation is almost 100%, 99.5 radioactively. So it's very, very specific. And I'm trying to envision this as the future of some of nucleic acid-based therapeutics. At the end, where are we going in carrier-mediated RNA therapy? I think everything that is listed here is correct. We need to avoid on-the-way mononuclear phagocytes unless this is our target. We need high specificity and functionality. We need a very fast internalization and burst release into the cells if we are delivering nucleic acid that needs to work inside. We need to improve the RNA drugs. For example, modified messenger RNA, still there's some issues. siRNA is more simple, and of course, genome editing is something very, very interesting, but less efficient. The efficacy now seems to be less working, needs to be improved. And then at the end of the day, I want to thank people in my lab, kind of international group of people, funding agency that help us. So you can see the ERC here is a very, very important compound. Also the NIH is very important, both the NIAID and the NCI. And I want to thank you for listening. And my last slide is the most important one, this one. You can see that I'm the only one who has a glass of wine. It means that I'm the only one who talks. I have a PhD, so right, I'm a philosopher, basically, and the rest really works. So thank you so much.
Thank you, Dan. Wonderful presentation. Any questions? Yes. Why am I not surprised?
Yeah.
So you heard me, right?
Yes,
Yeah.
I think I heard you.
Yeah. So
how
I-
it's
the
going to
question-
be cleared from the system,
Yes.
right?
How does it clear, and what are the side effects during the clearing process?
So I don't know the side effects because we haven't experienced any in non-human primates. This is the level that we are now looking at. Non-human primates, we haven't seen like cytokine induction, interferon responsive genes, nothing. I have to explain that we do all this packaging under a clean room condition. And we are using microfluidic system. We have a collaboration with a company in Canada named Precision Nanosystem that actually have microfluidic chips, and they're GMP. So if you do it under GMP raw material in a clean room, you could experience something. But what we were surprised that if we incubate this with like healthy humans blood samples, like PBMCs, peripheral blood mononuclear cells, we do not see induction of immune response to something like this, and not complement activation. Now, it's specific formulation and specific orientation. But if we do chemical conjugation, we do have complement activation. So this is another thing. We believe that the complement proteins are sitting on linkers areas. And in our case, since the antibody Fc is protected inside, they just see the protein like this in the binding orientation, and probably we will experience, I hope not, but probably we will, in a multiple dosing situation in humans, we're probably going to see some kind of immune response because there are no naive molecules. Always when you inject something, you will get something. The question is it going to be in the right direction or in the bad direction? So, for example, am I thinking about application in chronic inflammation? No. But in cancers, yes. Yeah. Yes. Yeah. But for us, it's a proof of concept because we had the antibody and we like the models and we are developing other strategies in Crohn's colitis, other antibodies and other fusion proteins. So for us, it was a proof of concept. The mantle cell lymphoma and other, I would say, more resistant blood cancers are my real target.
Dr. Sarma, your question.
Yes.
Your new linker system looks great. So, as you showed, if you bring down TNF-alpha, you won't get rid of the inflammation. You have to bring down also EL1 alpha, interferon gamma.
Right.
So my question is, have you tried a cocktail?
Yes. So we have tried, this is another. So we have tried the cocktail, and in IBD, there was a clinical study that suggested that if you're going to give IL-10, an anti-inflammatory cytokines, to the patient, it's going to be fantastic. And the clinical trials failed because the huge toxicity that the patient had, and nobody select those patients that has lack of IL-10. We can do it in mice because we have an IL-10 knockouts. And in mice, we can cure them. If we just bring them, for example, modified messenger IL-10, we cure them. But it's temporarily. So after a few days, it's a very long, it's a chronic model, then we start seeing something else is going up. So it's a very gentle inflammation, alpha inflammation, and I would be scared to try this in patient, even if a cocktail knocking down different things. I think cancer, for example, or some rare disease are more appropriate for this. Thank you.
And thank